This video will be covering A1.2 nucleic acids. Our first understanding is DNA as the genetic material of all living organisms. First, it is important to know that there are four major types of biological molecules: carbohydrates, lipids, proteins, and nucleic acids. Nucleic acids have two main functions. The first is to pass information between generations, and the second is to code for protein production. There are two types of nucleic acid: deoxyribonucleic acid and ribonucleic acid, which can be shortened to DNA and RNA. RNA is hypothesized to be the first source of genetic information to evolve, but today all living organisms use DNA as their genetic material. However, RNA viruses use RNA as their source of genetic information, but since viruses do not carry out all functions of living organisms, this does not count as an exception to the rule that all living organisms use DNA. Our second understanding is regarding the components of a nucleotide. A nucleotide is a monomer of a nucleic acid that contains a phosphate group, a deoxyribose or ribose pentose sugar, and a nitrogenous base. The carbon in the pentose sugar will be labeled anti-clockwise starting from here. And this diagram shows the basic structure of nucleotide that will be asked in the exam. A circle representing the phosphate group linked to a pentagon, and finally to a nitrogenous base, which can be represented using a rectangle. Our third understanding is regarding the sugar-phosphate bonding and the sugar-phosphate backbone of DNA and RNA. DNA and RNA have a backbone of repeating phosphate sugar units. The ends of these backbones are named 5' and 3'. The 5' end is a phosphate, while the 3' end is a pentose. Nucleotides join together to form nucleic acid polymers through a type of covalent bond known as a phosphodiester bond. This happens when the phosphate group of one nucleotide bonds with the sugar molecule of the next nucleotide. This creates a repeating sugar-phosphate backbone, which forms a structural framework of nucleic acids like DNA and RNA. The nitrogenous bases stick out from the backbone and carry the genetic information in the form of a sequence of bases. Nitrogenous bases can be one of four: adenine, cytosine, guanine, and thymine, which is found in DNA, while uracil is found in RNA. Based on the number of chemical rings in their structure, the nitrogenous bases are grouped as either purines or pyrimidines. Purines have a double ring structure. The order in which nucleotides are arranged in DNA or RNA serve as code for genetic information. A gene is a specific sequence of nitrogenous bases in DNA nucleotides that codes for the making of protein. Our next understanding states RNA as a polymer formed by condensation of nucleotide monomers. The RNA backbone is formed when nucleotides combine in a condensation reaction. A condensation reaction is a type of chemical reaction in which two molecules are combined to form a single molecule with the loss of a water molecule. Monomer nucleotides combine to form the polymer RNA chain. In this condensation reaction that forms RNA, the 5' phosphate group of one nucleotide forms a new covalent bond with the 3' carbon on the ribose of the next nucleotide. As a result, the chain goes from 5' to 3'. The sixth understanding states that DNA is a double helix made of two anti-parallel strands of nucleotides with two strands linked by hydrogen bonding between complementary base pairs. The DNA molecule is made of two long chains of nucleotides that twist around each other to form a double helix structure. These two strands are held together by hydrogen bonds between the nitrogenous bases. The complementary base pairing rule states that adenine always pairs with thymine through two hydrogen bonds, while cytosine always pairs with guanine. through 3 hydrogen bonds. This ensures the strands fit perfectly together. The 2 strands of DNA are also described as antiparallel, meaning that they run in opposite directions. One strand goes from 5' to 3', while the other runs from 3' to 5', which is important for DNA replication and function. Now we will look at the differences between DNA and RNA. DNA and RNA have some similarities, but their structures are quite different. DNA is double-stranded with deoxyribose sugar and the base thymine, while RNA, on the other hand, is single-stranded, contains ribose sugar, and uses uracil instead of thymine. Their functions are also different. DNA stores genetic information for the entire organism, while RNA helps carry out this information by making proteins. In terms of location, both DNA and RNA are found in the cytoplasm of prokaryotic cells because they don't have a nucleus. However, in eukaryotic cells, DNA is stored in the nucleus, while RNA is made in the nucleus but travels to the cytoplasm to help with protein synthesis. The 8th understanding looks at the role of complementary base pairing in allowing genetic information to be replicated and expressed. Complementary base pairing plays a key role in both DNA replication and gene expression. During DNA replication, each strand serves as a template for a new strand, because A always pairs with T or U in RNA and C always pairs with G. The sequence of bases is copied exactly, ensuring no genetic information is lost. Complementary base pairing is also important in gene expression. When DNA is transcribed into RNA, the RNA strand forms by matching its bases to the DNA template strand. This process ensures that the correct genetic code is carried to the ribosomes to make protein. DNA has limitless diversity because it is made up of 4 bases: adenine, thymine, cytosine, and guanine, that can be arranged in any order along the DNA strand. Even small changes in the sequence can create completely different genetic codes. The longer the DNA sequence, the more possible combinations there are, allowing for a huge variety in genetic information across all living organisms.